much, and firstly I'd just like to thank the conference organisers for the opportunity to speak to you today. Well, as my title slide suggests, I'm a registered Australia-New Zealand patent attorney, and I work in the chemical life sciences team at a company called Spruson & Ferguson. And prior to becoming a patent attorney, around 10 years ago, I worked in medical research. I did a PhD looking at antiretroviral drug resistance in HIV patients, and following that I was lucky enough to gain a postdoc over at the Pasteur Institute in Paris, and there I looked at immune responses in a cohort of HIV long-term non-progressors. So that's a little bit about my background, and here's an overview slide of what I'd like to cover in my talk today. Firstly, I'd just like to look at the connection between patents and the vaccine industry, and indeed the role that patents can play in bringing a vaccine through to market. And following that, I'll look at the concept of gene patents and the subject matter that is in gene patents and the scope of protection that those kind of patents actually offer. And that will lead into the discussion on Myriad gene patent litigation. There's been a lot of activity in that sense, particularly in the US and Australia in recent years, so I'll give a discussion on that and also touch upon a few additional cases that have occurred in the US recently which have had an impact on the patentability of certain biotechnology inventions. And finally, I'll look at some strategies that patent attorneys and others are taking in order to try and circumvent some of the rejections that the United States Patent Office is putting forward at the moment in relation to the patentability of some biotech inventions. So what's the connection between patents and vaccines? Well, I guess the number of patent applications relating to vaccines filed in a given calendar year is a relatively modest proportion of the total number of pharmaceutical patent applications, but nonetheless the numbers are still substantial, and you can see on this map here published by WIPO, which is the World Intellectual Property Organisation, that there are significant numbers of vaccine patents filed, particularly in North America and in China. So the basic tenet of any patent system is the principle of exchange. So in return for coming up with a new and non-obvious invention, the patent holder gets a 20-year patent monopoly over their invention, and in exchange the public gets a full and frank disclosure of that invention so that they can use it once the patent term expires. So I guess the relationship between patents and the vaccine industry is really one of incentivization, because we all know that to bring a vaccine through research and development and clinical trials and then actually get the product onto the market, it requires a lot of investment and resources. And companies simply aren't willing to do that if once their product gets to the market, someone can just go and copy it. So I guess the patents provide an incentivization for people to put the time and effort into developing medical products such as vaccines. And on the other side of the coin, there's, I guess, a long-running ethical debate as to whether patents serve to restrict access to important medicines and vaccines for people in third world countries, particularly who need them the most. So as a patent attorney, I'm well aware of the ethical debate surrounding patents, and in particular certain types of medical patents. And thankfully that's really not the focus of my talk today, but I thought I'd bring it into light in view of discussing gene patents, which is pretty close to that kind of debate. So what are gene patents? And I should probably point out before going into that, for those who aren't terribly familiar with patent applications, the monopoly given to a patent holder through a patent is defined in things called claims, and that's towards the end of the patent specification, and there are a series of statements which really set out the patent monopoly that's available to the patent holder. And generally there's two types of claims: composition of matter claims, which are generally directed to some sort of product, and a second category would be method claims, which, as the name suggests, would give you protection over a method of doing something, such as perhaps treating a disease or making a product. So a gene patent will typically have a composition of matter claim over a gene sequence. And what a lot of people don't realise is that offers a very broad scope of protection, because it doesn't just stop one synthesizing the gene, for instance, or including the encoded polypeptide in a product or a composition. It also actually stops anyone using that gene or its product for any purpose whatsoever. So you can picture a situation where a gene patent is filed and granted, and then 4 or 5 years later, a researcher realizes that the gene or its product has a very important role in a certain disease, perhaps for a diagnostic or therapeutic application. But because the composition of matter claim exists over the gene itself, no one's allowed to then develop that technology into a treatment or a diagnostic. So that causes a bit of angst, and people kind of, I guess, think that the scope of protection offered by gene patents is perhaps a little broad in comparison to the contribution made in the initial patent application. So what kind of claims might one see in a typical gene patent? Well, as I've mentioned, you're going to see a composition of matter claim directed to, say, a polynucleotide encoding the gene that's been discovered. And I guess the important thing to notice about that claim is that the preamble refers to an isolated polynucleotide. And the reason for that is that under any patent system in the world, you can't actually claim something as it exists in nature. So there has to be some distinction in your claim between the natural form of what you're claiming and what you're claiming in the patent application. And until recently, it's been a fairly accepted phenomenon throughout the world that if you refer to something that's isolated from its natural form, that's sufficient to move it into the realm of patentable subject matter. But that's now come under an increased scrutiny in the US, where there's been a lot of patent litigation on the basis of whether things that are isolated from nature are actually sufficiently removed from naturally occurring substances to actually confer patentability. So typically, if you're going to have a composition claim over a gene sequence, you'll also have a second claim over a polypeptide encoded by the gene. And generally, if you're at the point of filing an application over a gene sequence, you'll at least need to disclose some kind of use of the gene, because another patentability requirement is you can't just claim something or a product without any tangible use for it. So, for instance, you might know that the gene has some kind of diagnostic application, so you'd include a method of diagnosis by detecting the gene or its encoded product and correlating that with the disease state. Alternatively, if the gene or its product has therapeutic applications, you might include another kind of composition claim, a pharmaceutical that comprises the gene or its product with a pharmaceutically acceptable carrier, and then have a method claim that's directed to treating a disease by administering that kind of pharmaceutical composition that you claimed earlier. So in the concept of a vaccine patent, I mean, the claims that you might want to pursue are probably fairly similar in a lot of ways. You'd certainly want a composition of matter claim over your vaccine, because that offers the broader scope of protection and is perhaps the closest thing to a commercial product. So the type of composition matter claim you would have would really depend on the active component in your vaccine. So if it's a polypeptide, as I've shown claim 1 here, you would claim that as part of your composition. It might be an inactivated microorganism, such as by irradiation or otherwise, or alternatively an attenuated microorganism. So they're the sorts of composition claims you might see in a typical vaccine patent. And of course, when you're at the point of filing an application for a vaccine, you'll know that it's actually useful in inducing immunity against a certain disease or condition. So you'd have a method claim for preventing a disease in the subject or inducing immunity against something by administering to a patient the vaccine composition that you've disclosed. So that leads into gene patents, because the scope of well, particularly composition of matter claims in gene patents and indeed in vaccines is quite broad, and it brings a lot of angst in terms of ethical debate and whether patents should have that sort of, I guess, monopoly over important medicines and vaccines. And that's kind of crystallized a little bit in the recent Myriad gene patent litigation. So for anyone who's not familiar with the technology involved with the Myriad patents, it involved the BRCA1 and BRCA2 genes, and basically a suite of mutations in those genes is found to give a predisposition to either breast cancer or ovarian cancer. And I think the numbers were, I think, a 50 to 80% chance of developing breast cancer, or a 20 to 50% chance of developing ovarian cancer if you're detected to have some of these mutations in the BRCA genes. So scientifically it was an important breakthrough, and I think it was recognised as such by the courts, and the question was really whether it relates to a patentable invention or not. So firstly, looking at how the litigation went in the US, to give you a bit of background, the BRCA technology actually came out of a university originally, and Myriad as a company licensed the whole suite of patents on the BRCA genes, and that was sometime in the 90s. And I think towards the end of the 90s, Myriad became aware of another diagnostic company that was apparently developing breast cancer diagnostics based on the BRCA gene technology, and also a few researchers at NYU who were sort of developing, I guess, the technology in a way that Myriad viewed to infringe their patents. So they were fairly aggressive about it, and they set forth cease and desist letters and threatened legal action. And at that point, I believe the diagnostic company and the researchers ceased their activity. And effectively, Myriad, I believe, was the sole provider of these breast cancer diagnostic kits based on the BRCA gene technology, at least for around the next decade. But as they say, all good things come to an end, and they did for Myriad when there was a significant amount of litigation about their patents, which commenced sometime, I think, in 2009. And basically, the people challenging their patents did so on the ground that isolated gene sequences and similar claims don't relate to patentable subject matter under the US Act. So the first challenge was heard by the district court in the US, which is I guess a fairly low-level court, and it was quite surprising that the district court actually invalidated a lot of Myriad's claims. And I say that's interesting because basically it had been the norm in the US for quite a long time, and indeed around the world, that if your claim referred to isolated gene sequences, that was enough to put it into the realm of patentable subject matter. So for a fairly low-level court in the US to find otherwise and knock out a whole bunch of claims based on isolated gene sequences was probably a pretty bold decision. So predictably, Myriad did appeal that, and it went to the Federal Circuit Court of Appeals, who promptly overturned the initial decision of the district court and reinstated the claims and basically said that everything was valid in terms of claims towards isolated gene sequences. So finally it went to the highest level, which is the US Supreme Court, and that would be the final say. So everybody was waiting with bated breath, particularly in the biotechnology industry, to see what the US Supreme Court said. And basically they looked at the question fairly narrowly. Are isolated genes capable of patent protection according to the requirements of the US Act? And they held that they actually aren't. And it was basically, the decision was made on the pretext that the information encoded by a gene, once it's been isolated from its natural environment, so taken out of the genome or synthesized or whatever, is essentially the same as the information that's encoded by the gene as it exists naturally. So on that basis, they said, well, there's nothing patentable in that, and claims to isolated gene sequences are therefore invalid under US law. So this was, I guess, a fairly big decision, and it sent shockwaves throughout the biotechnology industry in the US and probably elsewhere throughout the world, because the US is a target market for most biotechnology companies. And it effectively wiped out about 4,000 to 5,000 currently issued US patents in one fell swoop, because obviously there are a lot of gene patents in force in the US. So it was a fairly profound decision. So in and around that time, the Supreme Court issued a couple of other important decisions that had an impact on the biotechnology industry, and unfortunately a negative one. And the most, I guess, prominent of those cases was the Mayo v. Prometheus case. And in that decision, they considered a patent which had a method claim, and basically the invention related to discerning an optimal dosage regime for a particular autoimmune drug. So the idea was to administer the drug in a certain amount, measure the amounts of a certain metabolite that's produced as a result of administering the drug, and then you correlate the two to I guess, come up with an optimal dosage for the particular patient. So it's sort of like a personalised dosage regime for this particular drug. And the Supreme Court sort of acknowledged that that was actually a good invention, and you know, it was very helpful to medical practitioners. But at the same time, they said it really relies on something as it exists in nature and a law of nature. So while it's helpful, it's just not in the realm of patentable subject matter, and the patent was subsequently invalidated. And similar sentiments were put forward in the Alice Court case, which I mentioned here, that actually related to a business method patent implementable by computer software, but the principles that the Supreme Court used in the US were the same, and effectively they invalidated that patent as well on the allegation that it lacked eligible or patent eligible subject matter. So the combination of those two decisions really put diagnostic patents in the biotech industry in a pretty bad space, because a diagnostic patent typically relies on making a correlation between the levels of a certain biomarker or analyte and a certain disease state. And based on the guidelines set up by the US Supreme Court in those two decisions, that relates to a law of nature, and it's not going to be patent eligible subject matter unless you have something more to distinguish the claim from nature. So I guess those sentiments were confirmed in this Sequinone case I've mentioned, the third one. That was actually handed down in June this year, and it was quite a good invention insofar as it related to, I guess, detecting genetic defects in fetuses and embryos, not by the usual invasive techniques, but by actually detecting circulating fetal DNA in the mother's bloodstream. So it was made much easier to actually find out whether embryos have genetic defects by virtue of this invention. So again, quite a good technology and a good advance over what was currently available. And the Federal Circuit Court also acknowledged that, but at the same time said that the claim itself relied on something that relates to a natural phenomenon. It's a law of nature, and therefore it can't be a patentable invention. So as it currently stands in the US, biotech patents that rely heavily on naturally occurring substances such as genes and maybe proteins, and also diagnostic methods are under fairly negative, I guess, assessment at the moment by the USPTO, which is the United States Patent and Trademarks Office. And that's reflected in the guidelines which the USPTO has issued, I guess, on several occasions as the court cases develop. And these guidelines are meant for patent examiners and patent applicants to determine whether the subject matter of their claims are actually patent eligible under the relevant provision, which is Section 101. And the guidelines have been widely criticised by patent applicants and patent attorneys, predominantly because they're very long-winded and difficult to decipher. They're very subjective, and in essence, they're not very pro-patentee. So it's brought a lot of angst to patent applicants, certainly in the biotech space. But to be fair to the PTO, on the other side of the coin, the Supreme Court decisions have kind of made it clear what things might not be capable of patent protection, such as isolated genes and diagnostic methods that rely on correlations between, I guess, analytes and disease states. But what they haven't really set out or given any real guidance on is how far you have to remove your invention from the naturally occurring phenomenon in order to establish a patentable position. So the PTO has been left with an unenviable task of trying to, I guess, come up with some guidelines that indicate that without really having any kind of knowledge themselves of how far you need to go. Well, the next few slides are on the Myriad gene patent litigation as it occurred in Australia, which has been interesting insofar as that up until recently it went completely the opposite way to what it did in the US. So Myriad's foundational BRCA1 patent was challenged in 2010, and the challenge was made by a breast cancer survivor pictured here called Darcy, and she was a member of a larger cancer advocacy group called Cancer Voices. And the original challenge went to a single judge of the Federal Court of Australia, and that judge really considered the sole question of whether a claim directed to an isolated gene sequence satisfies the relevant test under Australian law for patent eligibility, which is the manner of manufacture test. And to put it in a short way, the single judge of a federal court found that it did, and held that Myriad's claims to isolated gene sequences were patentable under Australian law as it stands. So Cancer Voices then appealed that decision to the full federal court. So that means that 5 federal court judges considered the matter, and they considered the same matter that the original judge did about isolated gene sequences, and also looked at the issue a little bit more broadly in terms of whether naturally occurring biological substances in general, once they're isolated from their natural environment, are capable of patent protection by virtue of satisfying this matter of manufacture test. So as the quote says here, they found that the structural and functional differences introduced by isolating a gene from its natural environment were sufficient to satisfy the requirements of this matter of manufacture test. And I guess some of those requirements were put out in a High Court decision many years earlier called the NRDC case, and the sorts of things you're looking at is whether the invention involves a requisite invention of man or the creation of an artificial state of affairs. And the full federal court ruled that those sorts of requirements were satisfied by the act of isolating something from its natural environment. So effectively, there are different considerations to what applies in the US, and perhaps that's a reason why the Australian court found differently to the US court. And indeed, the federal court actually referred to the US Supreme Court decision, and what they said reflected just that really the tests in the 2 different countries differ, and in Australia there's no need to consider really whether a composition claim relates to a product of nature or whether it's markedly different from something in nature. They're the tests that apply in the US, but they don't apply in Australia, so there's different considerations to make. So during all this time, there was also a group of prominent senators who sought to introduce a bill amending the Australian Patents Act to actually, exclude certain biological substances from patentability, particularly isolated genes and other isolated biological substances. And that bill never saw the light of day and was also rejected by Parliament. So we were in a pretty good position at that point where 6 federal court judges and the Australian Parliament had given a firm indication that isolated genes were patentable in Australia. And that left everyone feeling rather comfortable about it, even though the US situation wasn't going as well. So it was a surprise when in February this year the High Court granted leave to appeal the federal court's decision on the Myriad patents, and everyone was left wondering why the High Court would have done that when the decision at the federal court level was so unanimous, and even Parliament had put its hand up to, support the concept of patenting isolated genes in Australia. But nonetheless, the hearing went forward and occurred in late June this year, and we're expecting a decision probably towards the end of this year. And based on a conversation I had with a colleague who attended the hearing and reading some commentary on the hearing itself, it does appear that the claims in Myriad's patents that relate to isolated gene sequences could be on some shaky ground. So that's not a foregone conclusion in any way, but there is some possibility that those claims may be invalidated. And if that were the case, one could see that logically extending to other isolated biological substances such as proteins or biological compounds and otherwise. So we're kind of all waiting to hear what the High Court says on that decision, and it'll probably have a profound effect as it did in the US if it's a negative decision. So my final slide here today, I just want to discuss a couple of strategies that patent attorneys and others are using to try and address these problems that are arising in the US in relation to unpatentable subject matter. And I thought to discuss it in the concept of a vaccine patent, where certainly you're going to try and get a composition of matter claim over a vaccine. And I guess to be honest with you, if the vaccine or the antigenic component of the vaccine is a natural or mirrors a naturally occurring substance, so let's say it's just a protein from a microorganism or an antigenic determinant from a protein or maybe even a whole microorganism, if it's substantially similar to the natural form, you're going to strike problems in the US for a composition claim, because the USPTO is just going to say, well, it's not patent eligible, you're claiming something that's not different enough from what exists in nature. So there are a few strategies to try and get around that. People, obviously, the main one is to just modify your antigen in some way so that it doesn't reflect exactly what's in nature, and you can argue that therefore it relates to pattern or subject matter. Other people, I guess, combining the antigenic determinant with some other component, so you might have a complex or a conjugate, so maybe the other component assists in delivering the vaccine or enhancing immunogenicity or something like that. And obviously, if you're claiming a complex of that nature, it doesn't exist in a natural form, so that's another way of addressing it. Others are suggesting to include specific dosages of the antigen in your vaccine, because while the antigen itself may exist in nature, one can argue that the concentration of antigen in the actual patent claim doesn't reflect a natural state. And others are even using kit or package claims. So you claim the vaccine composition in the form of a kit or perhaps a syringe with instructions to use, and that's another kind of composition you can get around the actual vaccine that might be arguable doesn't relate to a naturally occurring substance. But thankfully, in the context of vaccines, you've always got the method claims to fall back on, and it's good in a way because the US Supreme Court decisions haven't indicated in any way that a claim that involves an active administration step to a patient, so a therapeutic method of treatment or vaccination, should be under scrutiny in terms of these Section 101 patent eligible questions. So a vaccine patent should always have a method of treatment claim where you're vaccinating using your vaccine composition. And even though that claim doesn't cover the vaccine composition itself, it really prevents anyone from using the vaccine for its intended purpose. So effectively, the scope of protection offered is still fairly broad by a method claim. Well, that's all I had time for today, and I'd be happy to answer any questions anyone might have.
Patent attorney Simon John Potter, thank you very much for this clarifying talk. Are there any questions?
Well, in terms of the US, I assume you're talking about. I mean, I should point out that in Australia, this sort of stuff isn't an issue at this point. Like, we're still in the same position, unless the High Court decides otherwise. But in the US, that probably wouldn't help you unless you actually put it in the context of a method of use claim. So the issue is really with, like, if you had a composition claim and just identified that there was a new use for the gene or a novel use for it, the PTO would say that's an inherent capability of what you're claiming. But if you put it in the context of a method claim, then potentially there's some way around it if you worded the claim in the right way.
Any more? Yes.
Comment on the situation in Europe.
The situation in Europe, well, in terms of this isolated gene scenario, it hasn't really changed as far as I'm aware. So it's fairly friendly for that sort of thing. I mean, you know, there's the standard types of objections that you'll have to go across or get around, but certainly there's no new case law that's indicating that a move or a shift against isolated genes at the moment. So I think that's pretty good. I mean, it's really the Myriad litigation. I think there's some movement in Canada as well where it might be adverse, but it's really restricted to Australia and the US at this point.
Yes.
So, in terms of biomarkers, you're saying the vaccine you can get around it by talking about modifications and things like that. Can you do the same with biomarkers? Like, it's a kit, therefore it comes as a package and things.
Yeah, that's.
Slow down biomarker discovery.
Well, yes, it's a real concern. I mean, there's several ways of approaching that, really, and yes, a kit is one way. Because in the US, if you provide a kit with components and instructions for use, the instructions for use are considered limiting on the kit as well. So, yeah, that is one way around it. You can, as we said, modify it, and some diagnostic applications work where you might have to administer a novel reagent in order that might bind the analyte or biomarker, and then you detect that. So if you can focus your claims in on the complex, which isn't naturally occurring because you're applying something to detect it, that's another way that you might be able to get a claim around it. So we're all trying to think up of these kind of nifty ways to get around it, but it's a bit difficult at the moment. And some people are even saying, well, look, this is a rapidly moving area in case law. I mean, you're getting quite a few decisions in the US about this sort of stuff every year. So people are just sitting on their hands and delaying prosecution in the hope that maybe over the next 1 or 2 years there will be some favourable decisions, and then they can move forward and try and get a more meaningful claim around their invention.
Again, thank you very much.